Okay, so what else has to happen during this process? So another protein we need to think about and understand is a protein called, for short, SSB, for single-stranded DNA binding protein. And this takes care of the problem that I mentioned before, which is if we go back to my illustration of a helicase, right? You can imagine if you just did this. I mean, this is perfectly base pairing to this, right? All these guys would base pair to one another, and they could immediately come back together, okay? And SSB actually helps to prevent that, although there's another thing that does that as well. So okay, so the first answer to this is actually quite simple, which is the leading strand polymerase rapidly replicates the single-stranded DNA behind the helicase. Okay, so you can see that there's only this little stretch here, and this may be very, very short, like 3 or 4 base pairs, before the polymerase is actually replicating that DNA. Okay, so that's going to keep those two strands from immediately coming apart. But the second is that the lagging strand template is rapidly bound by SSB. Okay, so what's SSB? SSB is, just so we have it all written out, the single-stranded DNA binding protein. Okay, in E. coli it's actually called SSB. In eukaryotic cells it's a 3-subunit complex called RPA. But importantly, in both cases, they have very similar properties. The first is it binds single-stranded DNA, but not double-stranded DNA with high affinity. All right. The second is it prevents strand annealing. And you might think, oh, well, that doesn't matter because the other strand is already replicated, so I don't need to worry about that. But just remember, before this is replicated, there's quite a bit of single-stranded DNA around, and you don't want that folding into some structure when there happens to be short regions of complementary DNA located. So for example, in a head-to-head repeat, you can actually get hundreds of base pairs of complementary DNA, and if that folded right up, it might make it more difficult to replicate. Okay? So SSB binds there, and the other thing is that SSB binds single-stranded DNA cooperatively. Okay, so what do I mean by that? I'll give you a simple illustration. Okay? So basically, what happens is that when SSB binds to one piece of DNA, SSB is much more likely to bind immediately next to another SSB. Okay? And then another, and then another, and then another, and it will basically rapidly fill all the binding sites on one single-stranded DNA before it really starts binding to others. Okay? And that's perfect at a replication fork, right? Because once you start putting SSB on a place, you can continue to rapidly expand that, especially on the lagging strand where you accumulate quite large regions of single-stranded DNA, up to a KB at a time. There's one other thing that's important about it, though, is that in the end, we want to replicate the DNA. And so it's important that it not only not interfere with replication, but that it rapidly come off in response to it. and that both are true. SSB bound DNA is readily replicated by polymerases. And structural studies suggest that the bases remain exposed when SSB is bound. Okay, so the most important thing for the replication machinery is to get access to the bases, and both the process of replicating it displaces them. Obviously, once it's double-stranded DNA, it no longer has affinity for that DNA. And secondly, the polymerase probably, although this have not been precisely mapped, there appears to probably be some direct interaction between the polymerase and the SSB that causes it to pop off the DNA. And that comes from studies where if you use an SSB from an unrelated species with a DNA polymerase, it doesn't work nearly as well as an SSB from the same species. Okay? No great mutants to identify those, but there's certainly an idea that that's true.